Ever stop to think, what happens if a really key blood vessel inside you gets damaged or blocked?
It's a critical situation, absolutely.
Yeah. And today on the deep dive, we're really going deep into this fascinating world of bioengineered vascular grafts, basically custom-made blood vessels.
Exactly. I mean, when your own vessels just aren't up to the job for repair,
Mhm.
maybe because of disease like atherosclerosis or, an injury,
Right.
doctors need a substitute. And traditionally, that's meant using one of your own healthy veins from somewhere else or using synthetic tubes.
And those have saved countless lives, no doubt. But they both have downsides, right?
They do. Your own veins might not be in great shape, or you might just not have suitable ones available. And the synthetic ones, well, they can have issues long term. Clotting, infection, sometimes they don't match the natural vessel's flexibility.
Which is why there's been this, long search in vascular surgery for the quote-unquote holy grail.
Mm-hmm. The ideal arterial substitute.
Mm-hmm.
Something that just works perfectly, integrates, and lasts.
And that's where bioengineering comes in, trying to meet that massive challenge.
It really is. And for our discussion today, we're leaning heavily on Rutherford's Vascular Surgery and Endovascular Therapy, the 10th edition. Specifically, the chapters covering bioengineered vascular grafts.
Great source.
Yeah, it gives a really thorough look at the history, the science behind it, and the actual clinical experiences, how it's played out in real patients.
Okay, so our mission for this deep dive, let's unpack the different ways scientists are trying to create these things, some really clever approaches.
Definitely.
We'll look at some surprisingly early history, key moments, and figure out where this tech is right now, and importantly, what it could mean for you or maybe someone you know in the future.
Sounds like a plan.
Yeah.
Where should we start? The history.
Yeah, let's do that. To really get where we are now, we need context, right?
Yeah.
The early days.
Absolutely. You have pioneers like Alexis Carroll, Nobel Prize winner, figuring out the basics of how to connect blood vessels back in the early 20th century. Foundational work.
And you mentioned a surprising name connected to him.
Right. Charles Lindbergh.
Huh.
Later in Carroll's career, they actually collaborated on developing techniques for organ preservation, sort of a very early precursor to the ideas behind tissue engineering.
Lindbergh. That's amazing. You think aviation, not perfused organs?
Shows how ideas cross-pollinate, doesn't it?
It really does. So while using a patient's own vein, the saphenous vein, has been around a long time. It wasn't until, the 1950s or so that synthetic materials really took off. Dacron, PTFE.
Exactly. Dacron for the bigger arteries, PTFE or Teflon, often for smaller ones, those became workhorses.
But what's really wild is that the idea of biological grafts wasn't completely absent even then.
Not at all. It's fascinating. Back in the 1940s, you had Robert Gross and his team doing experiments, transplanting aortic tissue in dogs.
1940s, wow.
Yeah, and a key thing they figured out was about preservation. Simple freezing wasn't great, but storing the tissue carefully in nutrient solutions just above freezing worked much better, like for weeks.
That really laid the groundwork for, tissue banking later on, didn't it?
It absolutely did. And this wasn't just dogs. They moved fairly quickly to using modified arteries from human cadavers.
I mean, people.
Yes, to fix aorta problems in kids, even for creating shunts for certain heart defects. So the potential of modifying donor tissue was clear very early on.
But obviously, that approach had its limits. Scalability, predictability.
Exactly. But it established the core concept, using intact isolated blood vessels from donors, but treating them somehow.
Okay, so let's dig into that first category then. These intact, isolated blood vessels. What are we really talking about here?
So the basic idea is you take a whole blood vessel from a donor, could be an animal, which is xenogeneic, or another human, allogeneic, and you process it a lot.
To stop the body rejecting it.
Precisely. To minimize the immune response. Even though they start with donor cells, the processing is so extensive, they really fall under the umbrella of bioengineered grafts.
Okay, let's start with the animal ones, xenogeneic. What's been used?
Well, one of the oldest and maybe best known is artegraft.
Yeah.
It's derived from cow carotid arteries.
Bovine carotid.
A cow artery. Yeah. And the process involves using enzymes to get rid of most of the cellular stuff, and then chemically fixing the remaining collagen structure with something called glutaraldehyde.
Fixing, like preserving it.
Sort of like tanning leather, in a way. It makes the tissue stronger and, crucially, much less likely to trigger that immune rejection.
And this has been around a while.
A long while. FDA approved back in 1970.
70?
Wow. Primarily for creating access points for hemodialysis, but also used for bypasses in the legs, sometimes as patches. Still used today.
Half a century of cow arteries helping people. That's pretty incredible. Any drawbacks?
Well, yeah. There have always been some concerns about calcification over time, calcium deposits building up in the walls.
Making it stiff or brittle?
Exactly. That can lead to weakening, potentially aneurysms down the road.
Uh,
Another animal option is Procol.
Procol? What's that from?
That one comes from bovine mesenteric veins from the cow's gut area. It also gets decellularized, fixed with glutaraldehyde, and even hit with gamma radiation.
Wow, quite the treatment. Why veins?
The thinking was that veins are naturally more elastic, more compliant than arteries, and maybe that compliance would be an advantage, especially for dialysis access where flow dynamics are tricky. It got approved in 2003, specifically for dialysis patients where other grafts had failed.
Did it live up to that hope? Better compliance, fewer blockages?
Some early studies showed okay patency rates. That's, how well the graft stays open.
Right, blood flow.
Yeah. But it hasn't really become a go-to graft across the board. Limited widespread use.
Okay, so animal sources have limitations. What about using human donor vessels, allogeneic grafts?
This seems more intuitive, maybe. Using human tissue for humans.
Yeah.
But the early processing methods used for animal tissues didn't translate well, especially for human arteries, which have their own tiny blood supply. But then came improvements in cryopreservation, really controlled freezing plus fixation methods.
And that led to?
That led to the cryovein graft, developed by CryoLife. It's a human vein, processed, and it's approved for both dialysis access and peripheral bypasses.
Okay, so a preserved human vein. Advantages?
Well, a big perceived advantage is better resistance to infection compared to purely synthetic materials. So if a synthetic graft gets infected, this might be an option.
Makes sense. Downsides?
Still some potential issues. Even with processing, there can be concerns about blood type compatibility and the recipient developing antibodies against the donor tissue. That could be a problem if they later need, say, a kidney transplant.
Ah, right. Cross-reactivity.
Yeah. And of course, relying on human donors always brings up questions of supply, though veins are maybe less critical than organs.
Got it. So these intact vessels were a step, but maybe not the final answer, which takes us to luminal modification, biohybrids. Sounds like mixing things up.
That's a good way to put it. It's realizing how vital that inner lining of our blood vessels is, the endothelium.
It's not just a pipe, right?
Not at all. It's super active, controls clotting, blood flow, inflammation, everything. So the idea with biohybrids was, can we take a synthetic graft, like EPTFE, and make its inner surface act like a real endothelium?
How would you even do that?
The main approach, starting back in the late 70s actually, was to take the patient's own endothelial cells.
From where?
Usually harvested from a small piece of vein. Then you'd grow these cells in the lab and seed them onto the inner surface of the synthetic graft before implanting it.
Seeding them, like planting them.
Pretty much, yeah. Trying to get them to form a living layer. There were single-stage methods done during surgery and two-stage where cells were cultured for weeks first. And some studies in the 90s looked really promising. PTFE grafts seeded like this showed patency rates in leg bypasses that were almost as good as using the patient's own saphenous vein.
That sounds fantastic. Why isn't this standard practice then?
Well, the practicalities were tough. Getting enough healthy cells from the patient wasn't always easy. And then, the bigger problem was ensuring those cells actually stuck to the synthetic surface and survived and functioned long-term inside the body.
Right. They might just wash away or die off.
Exactly. So despite some early excitement, it just didn't translate into widespread, reliable clinical success.
Did they try to improve it?
Yeah. Later work looked at conditioning the seeded grafts in the lab before implantation, exposing them to physical forces, like pressure pulses and fluid flow shear stress, trying to mimic the environment inside an artery.
To make the cells tougher, more adherent.
Kind of, yeah. To encourage them to organize properly and hold on better. There was some evidence this helped, but overall, the complexity, the cost, the reliability, it didn't quite get there for routine use.
Okay. So modifying existing things was hard, which leads us finally to building new vessels from the ground up. Tissue-engineered vascular grafts, TEVGs. This feels like the real cutting edge.
It absolutely is. TEDGs are a totally different philosophy. The idea is to create a conduit that starts out cellular or tissue-based, maybe with a temporary scaffold.
A scaffold, like scaffolding on a building.
Exactly like that. A temporary support structure. The key goal is that this initial structure, especially if it's synthetic, degrades over time and gets replaced by the patient's own cells and tissues.
So the end result is?
A completely living, functional blood vessel. made by the patient's body, essentially, with a natural lining, able to remodel, maybe even grow. That's the dream.
That is the dream, a living replacement. So how do you actually make a TEVG? Sounds like there are a few ways.
Broadly, yeah, we can think of maybe three main categories. First, grafts created in vivo, basically using the body itself as the factory, the bioreactor.
Okay.
Second, scaffold-based TVGs, where you grow cells on that support structure, usually in the lab.
Got it.
And third, sheet-based TVVGs, where you grow cells in layers like sheets and then assemble them into a tube.
Let's take the in vivo bioreactor first. Using the body, how does that work? Sounds a bit sci-fi.
Well, the earliest attempts were pretty clever, actually leveraging the body's foreign body reaction. There was this thing called the Sparks mandrel technique back in the 60s.
Stark's mandrel.
Yeah. You'd implant this rod or tube, maybe silicone or Dacra covered under the skin, often in the thigh, and you just leave it there for weeks. And the body would form a thick layer of connective tissue all around it, trying to wall it off. Then you'd surgically remove the original mandrel, leaving behind this tube of the patient's own tissue.
And use that as a blood vessel.
That was the idea. Pretty ingenious, right? But a bit crude.
Did it work well?
Not really. They had lots of problems. Poor patency, they blocked up easily. They could form aneurysms, get weak. Plus you had to wait weeks and weeks and then do another big surgery. It kind of fizzled out by the 80s.
Okay, so that specific technique didn't pan out. But the idea of using the body's response, did that evolve?
It did. Later researchers tried variations, like implanting celastic tubes in the abdominal cavity, the peritoneum of animals. And that actually generated a more structured tissue tube lined with mesothelial cells.
Better result.
In animals, yes.
Yeah.
But translating it to humans was still tricky. Long incubation times, multiple surgeries. Not ideal.
So then the idea became maybe use an animal as the bioreactor for a human graft? Is that where OmniFlow comes in?
Exactly. To get around that long wait time in the patient, they tried implanting silicone tubes under the skin of sheep.
Sheep, okay.
After a few months, these tubes get coated in a thick layer of sheep collagen and other matrix stuff. Then you harvest that collagen tube, process it, sterilize it, maybe reinforce it.
Reinforce it how?
The later version, OmniFlow 2, incorporated a polyester mesh into the wall during the sheep phase, giving it much better strength, preventing aneurysm.
So it's used in people?
Yes. It got approvals in several countries, mainly for hemodialysis access. And studies showed its patency rates could actually approach the gold standard AV fistulas, and crucially, with lower infection rates than synthetic PTFE grafts.
That sounds like a real step forward, using the biological process, but indirectly.
It was a significant step, yeah.
Yeah.
More recently, people are exploring ways to modulate that foreign body response more precisely, using different template materials or pretreatments to grow grafts in situ again, but maybe faster and better. Still research, though.
Okay, fascinating. Let's move to the second big category, scaffold-based TEVGs. This sounds more engineered, more controlled.
It is. This is mostly in vitro work happening in the lab. You could argue the real pioneers here were at MIT in the 80s, Eugene Bell and his group.
What did they do?
They created the first prototype engineered vessel. They mixed collagen with smooth muscle cells and cast it around a tubular mold. Showed it was possible to grow something vessel-like in a dish.
But is it strong enough?
That was the catch. Not really. It needed external support, couldn't handle blood pressure on its own. But as proof of concept, hugely important, it actually led to the start of a company, Organogenesis.
So the big hurdle became making scaffolds that were strong, but also biodegradable, something the body could take over.
Precisely. And that's where work by people like Joseph Vicanti and Robert Langer comes in. They were pioneers in using biodegradable polymers for tissue engineering generally. For blood vessels, researchers like Christopher Brewer and Tosharu Shinoka started working with polyglycolic acid, PGA.
PGA. You said that's like dissolvable stitches.
Exactly the same stuff. It breaks down in the body over time. They first used it to grow heart valve leaflets in lambs.
And this led to the very first clinical use of a TVG, right? In a person.
Yes, a landmark moment. In 2001, Shinoka's team reported using a TEVG to reconstruct a pulmonary artery in a child with a congenital heart defect.
How did they make it?
They took a biodegradable scaffold made of PGA and another polymer and seeded it with cells isolated from the patient's own bone marrow. Grew it for a bit, then implanted it.
And the significance?
Huge. It showed a lab-grown vessel could actually function in a human. And because it used the child's own cells and a dissolving scaffold, there was hope it could grow with the child.
Avoiding repeat surgeries, that's massive for kids.
Absolutely. And they had success in several more pediatric cases. But these grafts weren't strong enough for high-pressure systemic arteries like in your leg or arm. And there were some worries about inflammation as the PGA broke down.
So strength was still the key issue for wider use.
Strength and integration, yeah. So researchers went back to improve that. Laura Nicholson, working with Langer, made big strides.
Wow.
They figured out that if you grow the vessel cells on the scaffold under pulsatile conditions in the lab, mimicking the pulsing pressure of blood flow.
Like exercising the growing vessel.
Exactly. It made the cells organize better, produce more matrix, and a resulting vessel was much, much stronger. They also perfected methods to decellularize these engineered vessels.
Decellularize. Take all the cells out. Why?
To get rid of the donor cells. If you grow a vessel using cells from donor A and put it into patient B's immune system will attack it. By removing all the cells, you're left with just the extracellular matrix, scaffold, the collagen, elastin, etc., which is much less likely to cause rejection.
So you create the structure, then make it immunologically blank.
Precisely. And interestingly, their work suggested maybe you didn't even need to reseed it with the patient's own cells before implantation. The idea emerged that the patient's own cells might just migrate into the cellular scaffold once it's in the body.
And this thinking led to the humacyte vessel, the HAV.
Yes, it did. Humacyte really built on that. They used smooth muscle cells from human donors allogeneic, so not matched to the recipient, grow them on a biodegradable PGA mesh scaffold in vitro under those pulsatile conditions for weeks.
To build up the strong matrix.
Right. Then they do a very thorough decellularization process. What's left is this human acellular vessel, the HAV. Basically, a natural scaffold made of human collagen and other proteins, but no cells.
And the advantage?
Lab tests show it's strong, compliant like a real artery. And crucially, preclinical trials in baboons showed they could implant these HAVs without needing any immunosuppression drugs, and they stayed open.
No rejection.
Seems like the decellularization really works to overcome that immune barrier. It's a big deal.
An off-the-shelf, strong, non-rejectable human vessel framework. Wow.
Yeah. And similar decellularization ideas are being applied to things like heart valves, too, trying to create better, more durable replacements.
Okay, quickly, what were the other scaffold ideas? Xenogeneic and human tissue scaffolds.
Right. Xenogeneic scaffold means taking an animal vessel, say a pig aorta, decellularizing it, and then trying to seed that scaffold with human cells from the patient, kind of humanizing the pig scaffold. Still mostly experimental.
And human tissue scaffolds.
This was pioneered by Michael Acheson. They took a deceased human donor iliac vein, decellularized that, and then seeded it with endothelial and smooth muscle cells grown from the recipient's own stem cells.
A fully human, personalized scaffold.
Exactly. They used this in one really remarkable case for a child with a major vein clot, a groundbreaking proof of concept.
But think about the logistics.
Yeah. Finding a suitable donor vein, isolating stem cells, growing them, seeding the scaffold takes a long time, hard to scale up.
Precisely. Donor availability, time, cost. Major hurdles for that approach right now.
Okay, last category. Tissue-engineered sheets, building layer by layer.
Right. Instead of seeding cells onto a preformed scaffold, you grow the cells, smooth muscle cells, fibroblasts, into concluent sheets in culture dishes.
Like a sheet of living tissue.
Yeah. Then you carefully lift these sheets and wrap them, layer by layer, around a temporary tube or mandrel. You let them fuse together to form a vessel wall.
Does that work? Is it strong enough? Early attempts struggled with structural integrity. But Nicolas Leroux and his group developed a pretty sophisticated process, using cells from human umbilical cords, smooth muscle cells and fibroblasts for the wall, and endothelial cells for the lining. They managed to create a completely biological human vessel this way that was strong enough.
Any downsides?
The main one was time. It took like 4 to 6 months to manufacture one vessel. Very labor-intensive.
And this led to a clinical product, Cytograft's Lifeline.
Yes. Cytograft commercialized this sheet-based technology, creating the Lifeline graft using the patient's own cells, autologous. It was targeted for hemodialysis access.
How did the trials go?
Preclinical looked good, but the early phase 12 clinical trial, well, it had a pretty high early failure rate. They later figured out it was likely due to using some bovine proteins during the manufacturing, which caused an immune reaction in patients.
An unexpected complication.
Yeah. They also saw some issues with the grafts dilating or stretching out over time, and again, that super long manufacturing time, 24 weeks, was a major practical barrier. Not exactly off the shelf.
Did they try other things?
They experimented with freezing the autologous vessels to store them and also making sheets from allogeneic donor cells and decellularizing those to try and improve availability, but that product hasn't really moved forward clinically in a big way.
Okay, so we've covered a lot of different approaches. Let's talk clinical reality. How have these actually performed in patients, starting with the intact, isolated vessels?
Right. Artegraft, cryovein, they've been used for decades, mainly for dialysis access, sometimes peripheral bypass. Patency rates, often comparable to synthetic grafts in those roles.
But not perfect.
No. Durability is a question. We mentioned calcification risk with artegraft, potential immune issues or sensitization with cryovein. Infection rates might be lower than synthetics, which is good, but they aren't zero. Procol, the bovine vein one, just hasn't gained much traction.
And the biohybrids, the cell-seeded synthetics.
Despite that early promise, larger trials generally failed to show a consistent, significant benefit. The technical challenges just seem too great for routine use. Limited success, really.
Which brings us to the fully tissue-engineered grafts. What's the clinical scorecard there?
Well, Shinoka's pediatric pulmonary artery work using biodegradable scaffolds seeded with autologous cells has been a real success story in its niche. Good long-term patency reported, potential for growth, and if they narrow, sometimes they can be treated with balloon angioplasty. But again, not strong enough for adult systemic pressures.
Cytograph's lifeline.
As we said, hit those early hurdles with immune reactions and the long manufacturing time. Not currently a major clinical player.
Olafson's single case with the seeded donor vein.
Incredible science, amazing proof of concept that it's possible, but just not scalable with current methods.
Which leaves Humacyte's HAV. That seems to be where a lot of the current buzz is.
It really is. The clinical trial data coming out is quite promising. Phase 1 and 2 trials for hemodialysis access, often in patients who can't get a standard AV fistula, showed the HAV was well tolerated.
Tolerated meaning?
Low complication rates, very low rates of infection, which is huge for dialysis access. And maybe most importantly, biopsies showed that the patient's own cells were migrating into the cellular graft over time.
So it was actually becoming living tissue, repopulating.
Exactly.
Yeah.
Evidence of remodeling, development of an endothelial lining, and importantly, follow-up out to 5, even 6 years in. Some patients show no aneurysmal degeneration. It seems to hold its structure.
That's key for long-term use.
Absolutely. Phase 3 trials are ongoing now, directly comparing HAV to synthetic grafts and even fistulas for dialysis access. These could really change practice if the results hold up. Think about the impact on patients dealing with repeated graft failures or infections.
Huge potential impact. And you mentioned peripheral bypass, too, leg arteries.
Yes, a phase 2 trial done in Poland used the HAV for femoropopliteal bypass, basically, bypassing blocked arteries in the thigh or behind the knee. The 2-year results look good. Decent patency rates, low complications, no amputations in that group, and again, no signs of immune rejection.
So the evidence is building for this cellular, off-the-shelf approach.
It seems to be. The ability to have a readily available, strong biologic graft that integrates and becomes living tissue without needing immunosuppression. That's a very attractive combination. But, we still need the results from these larger, longer-term phase 3 trials to really know how it stacks up against the current standards in different situations. Durability across diverse patient populations is still the big question.
Right. It sounds incredibly promising, but clearly no magic bullet yet. There are still limitations, risks across the board.
Absolutely. We've touched on them. Patency and durability concerns for the older isolated vessels, technical hurdles for biohybrids, and for the TVGs, even the promising ones like HAV, we need more long-term data from large trials to really understand cost-effectiveness, true long-term durability, and performance in all patient groups. Any new technology carries potential risks until it's widely studied.
Okay, looking forward then. What's next on the horizon, and what future advances might push this field even further?
Well, 2 technologies get mentioned a lot. First is 3D bioprinting.
Printing blood vessels.
Potentially, yeah. The idea is to use bioinks, mixtures of cells and biomaterials, and precisely print complex, layered vascular structures. It could allow for custom shapes, maybe even incorporate branching patterns.
Tailored to the patient.
That's the hope. It's still early days, lots of challenges with getting the right materials, keeping cells alive during printing, achieving strength, but it's developing fast.
Okay, what's the second one?
Electrospinning. This is a technique that uses electrical fields to draw out incredibly fine fibers from polymer solutions, or even solutions containing cells.
What does that achieve?
You can create scaffolds with a structure that more closely mimics the natural extracellular matrix of a blood vessel wall, the alignment, the porosity. It could lead to scaffolds that integrate better, have better mechanical properties, maybe even combine electrospinning with bioprinting.
Just so better building blocks, essentially.
Right. Finer control over the structure at the micro level. But again, scalability, long-term biocompatibility, cost. These are hurdles to overcome before clinical reality.
It's incredible progress, though. So wrapping up this deep dive, what's the main takeaway for you?
I think it's the sheer progress over, 50-plus years?
Yeah.
From those early attempts with preserved donor vessels, which was innovative for its time,
Right.
through the challenges with synthetics and biohybrids, to now, where we have multiple ways to create tissue-engineered grafts. And crucially, the demonstration, especially with the cellular approach like HEV, that it seems feasible to create strong, non-immunogenic, potentially off-the-shelf biological tubes that the body can adopt and turn into log tissue.
It really feels like we're moving beyond just patching pipes to actually creating biological solutions.
That's a great way to put it.
Yeah.
The focus now in ongoing research and trials is refining these technologies, improving manufacturing, reducing cost, ensuring long-term biocompatibility and durability, understanding thrombogenicity, all of the things needed for widespread clinical use.
The future looks optimistic, then, for potentially millions of patients dealing with vascular disease.
I think so. The journey, as you said, has been long and complex. But these recent advancements in bioengineering, they really do offer incredibly promising solutions. It feels like a potential revolution in how we treat vascular disease is getting closer.
Definitely. Which leads to maybe a final thought for you, our listener, to consider.
As these bioengineered vessels get more sophisticated, maybe they truly become living tissue within the recipient, remodeled by their own cells. What does that mean long term? Are there unforeseen biological interactions? How might they respond to future conditions or diseases in the body decades later?
Yeah, that's a fascinating ethical and biological question, isn't it? Where does the graft end and the patient begin over time? It's a space where science, engineering, and our deep understanding of the body are all pushing forward together. So if this deep dive into bioengineered vascular grafts has caught your interest, definitely look into the ongoing clinical trials, the research happening. It's just an amazing example of human ingenuity tackling really tough healthcare problems.
Couldn't agree more.